Hello, my name is Vikas. I'm a faculty in the Chemical Engineering Department here at Kansas State University. Today I'm going to talk about graphene interfaced with biological cells and the opportunities and challenges associated with it. So this is a slide which shows the gist of what I will be talking about today. In this image here, there is a biological cell which is sitting on a graphene sheet which is attached to two electrodes, one which is supplying holes, other one is supplying electrons, and the bacterial cell or the biological cell is interfacing with the graphene and changing its electrical properties, and the change in the electrical properties then gets converted into a change in the electrical signal which is detected and provides information on what is happening on the bacterial surface or the biological surface. So let me first start talking about what graphene does and why graphene is so sensitive. Graphene is a single atom thick sheet of carbon atoms linked to one another by sp2 bonds. What that does is it produces an electron cloud which is confined in a single atom thick region. When a bacterial cell or a biological cell sits on the surface of graphene, the surface has dipole moments has dipoles. These dipoles have electric fields. These electric fields then impinge on the surface of graphene and changes the electrical properties. Now, when a bacterial cell sits on any other surface, for example, the surface of this table here, it will interact with the top atoms on the surface of this table. Now, when they interact, they will change the electrical properties of those top atoms. However, the effect will be averaged over the thickness of the tabletop. Because of this averaging, the net effect will be negligible. So what graphene provides is extremely thin structure which leads to a very high sensitivity. Why? Because it is the thinnest material in the world, just one atom thick. Some more reasons why graphene is so sensitive are listed here. The first one is the quantum capacitance. So graphene has a high quantum capacitance which acts as an amplification factor. So anything that happens on the surface is amplified. Any electric field is amplified. The carriers are confined within a single atom thick structure. This is what we just discussed. Because of the small thickness of graphene, the averaging is negligible, which means that the whole effect is transferred to the electrical properties of graphene. It has high carrier mobility. The carrier mobility is the speed with which electrons and holes move. So any signal which is generated from the interfacing is transferred at high speeds because of the high carrier mobility. It has large surface area, and this is important for applications with cells. Why? Because cells are large. So the bacterial cells, mammalian cells, these have large surface areas, and for interfacing with these large surface areas we need a material which has large surface area, and graphene has that. Lastly, graphene can be easily functionalized, which can add specificity to graphene. So one of the first works that we did in 2008 was to use graphene to make biomolecular sensors. We made a sensor for DNA, and this was a paper which was published in Nanoletters in 2008. Here we functionalized graphene with the DNA molecule, and then we brought in a complementary DNA which interfaced with the DNA attached to graphene, and that led to a change in the electrical properties. And so this is a DNA sensor which is highly sensitive and also specific. The specificity is because of functionalization, the sensitivity is because of graphene itself. We use the same principle to make a bacterial sensor, in which we attached a bacterial cell on graphene, as you can see over here. So you have a bacterial cell which interfaces with graphene, and as after attachment of the cell, the conductivity of the graphene device increases, and so one can make a graphene-based bacterial sensor, which we showed in 2008. This image over here shows us the specific attachment of bacterial cells with graphene surfaces, and as you can see over here, most of the bacterial cells are going or depositing only on graphene surface, and this is because of the specific attachment of the bacteria on functionalized graphene.
My name is Phong Nguyen. I'm a research assistant from Dr. Vikas Berry Group. Today I'm going to talk about the challenge in this field, which is graphene is too specific, which means it interacts with any molecules that come near to its surface. One way to increasing the specificity of graphene is to covalent bond of graphene with the specific molecule. The problem is that once we do that, it deteriorates the graphene electrical properties. An alternative way to increasing the specificity of graphene is through pi-pi interfacing. In one of our experiments, we conduct the pi-pi interfacing of graphene with neutravadine and PCA complex. In this complex, we also introduce a PCA, which is a dye molecule, which help us to observe under the confocal microscope. The below picture shows us that the changing or the increasing in the contrast, which represent the pi-pi interfacings.
So graphene biosensors, or graphenes interfacing with biological cells, can have several applications. Some of them are high sensitivity biosensors. So the high sensitivity of graphene can be employed to detect biological cells, as we showed. Also, graphene can be interfaced with single cells to study biophysical phenomena happening on its surface. Another interesting application that can evolve from these studies is production of biocellular batteries, where we can use some bacterial cells like Geobacter, which produce protons on their surface, and we can extract these protons by interfacing these cells with a PN graphene junction. And in having a PN graphene junctions, these protons produced on the surface could be made to go in one direction while the electrons go in the other direction, and we could have a charge separation to produce a battery. So a Journal of Physical Chemistry has allowed access to new knowledge for several years. It has recently produced some really high impact articles on graphene science and technology, which have received heavy citations. Also, our work on microwave functionalization, which was published in Journal of Physical Chemistry Letters, has received tremendous attention.